Okay, let's try and finish out at least chapter 17. We've got 2 chapters left. Getting close to the very end of all of this. So right now we're going to be talking about transcription or synthesis of RNA. So if you recall earlier, the previous chapter we talked all about DNA and RNA, kind of what makes them up, as well as the replication of DNA, of DNA itself, and how it's wrapped around the histones, and how we need to acetylate or methylate as well different parts of the DNA and the histones in order to express or repress specific genes. We also talked a little bit about the different types of mutations and just touched on how those mutations are also fixed. You will see a lot of that information pop back up in future trimesters. I'm not positive right now exactly which classes that those do pop up, but I do recall those all coming back several times. So make sure you spend the time to go through that material. Okay, so now chapter 17, transcription, synthesis of RNA. So now we're going to be talking about, so when a gene is being expressed, so when those histones have been acetylated and it's open and we have a gene being expressed and our little CG islands are not methylated and not repressing the gene, what actually happens? Well, we get transcription or synthesis of the RNA. So we're going to describe the structure of RNA, list the functions of the different types of RNA, and also describe transcription in the eukaryotic cells. So ribonucleic acid is a polymer of purine and pyrimidine ribonucleotides. So notice here the big difference here is that rather than thymine, we have uracil. The uracil takes the place of the thymine. One pyrimidine base in DNA and RNA are different. The RNA contains uracil instead of thymine. Thymine is present in DNA. During synthesis of RNA, joining of the ribonucleotides occurs from the 5' to 3' direction, just like in DNA. Each ribonucleotide is joined by a phosphodiester bond, just like in DNA. The 3' OH group of the first ribonucleotide joins with the 5' phosphate group of the second incoming ribonucleotide, exactly the same as DNA. RNA is a single-stranded molecule. This is where it is now different. RNA is a single-stranded, DNA is double-stranded. However, if the ribonucleotide sequence in RNA contains appropriate complementary base sequence with opposite polarity, the single-strand RNA can form wording's a little weird there. The RNA can fold on itself, acquiring double-stranded structural characteristics. So it's not fully double-stranded like DNA is, but it can kind of fold in on itself and kind of have and end up with some of those double-stranded characteristics as long as it's the different ends of it are complementary and they can match up. So you can have hairpin loop structures, we'll get into those, where the bases within do not match for base pairing. But unlike DNA, RNA molecule does not necessarily follow Chargaff's rule, i.e. it does not contain equal number of adenine and uracil or guanine and cytosine residues. That is also because a lot of it is single strand, or parts of it can be complementary, but not all of it necessarily is complementary. You will not follow those same Chargaff's rules. Remember, Chargaff's rule is stating that the numbers are connected. So adenine equals the amount of adenine is equal to the amount of thymine, the amount of guanine is equal to the amount of cytosine. Okay, so RNA structure, very, very similar to DNA. We have a ribose sugar. Difference is that DNA is a deoxyribose. Now we just have a ribose sugar. We have a phosphate. We have a purine base, adenine or guanine, and a pyrimidine base, uracil or cytosine. Again, here's all of our pictures of all of those stuff. So RNA is single-stranded. When strand loops back on themselves, the bases on opposite sides can form base pairs. So adenine forms 2 hydrogen bonds with uracil, and guanine forms 3 hydrogen bonds with cytosine, so very similar to DNA. And it forms secondary and tertiary structures important for RNA function. So RNAs recognize DNAs and other RNAs by base pairing. Some RNAs may have palindromic structure. palindromic structure refers to a sequence having inverted repeats or base sequences. Palindromic sequences produce hairpin-like structures. We'll see a picture of that in a little bit. One example to give an idea about palindromic sequence can be described as the word rotator. If you read the word from left to right or right to left, it is identical. Palindromic sequence can be present in both DNA and RNAs. It forms a two-fold symmetry as it reads from 5' to 3' end or 3' to 5' end. Here we show one example of palindromic sequence in RNA, which produces a double helix-like structure and also can form a hairpin loop where the base pairing is not matched. So here we have this palindromic-like structure, and so these guys will match up, but this structure here does not, so it forms a hairpin loop. Let me actually see if there's a better picture. These are better pictures, a little bit of these hairpin loops, kind of what it looks like, where you have the bases pairing and the connected here, and then you get hairpin loops where these do not actually bind together, so they form these loops. Okay. Make sure I didn't skip anything. Yep, good to go. Again, we have the same phosphodiester linkages, just like in DNA. Some RNAs are biocatalysts, so ribozymes. Biocatalysts, remember catalysts speed stuff up, they lower the activation energy. Ribozymes usually are precursors of ribosomal RNA or rRNA. Catalytically removes internal segments of themselves, splicing the ends together. They act as ribonucleases, cleaving other RNA molecules. So that is their function. So that when you see ribozyme, this is their function. They cleave other RNA molecules. They contain peptidyl transferase activity, which is an enzyme required in protein synthesis, and are composed of RNA. So let's look at the different types of RNAs. We have ribosomal RNA or rRNA, which is associated with proteins to form ribosomes. Messenger RNA, the mRNA, contains a poly A tail at the 3' end and a 5' cap containing 7-methylguanosine. These are synthesized from a specific DNA sequence. And we have transfer RNA, which is a cloverleaf structure which contains 3 nucleotides anticodon specific for amino acid codons. So our transfer RNA is going to be responsible for bringing and carrying our amino acids. Messenger RNA is the kind of like the copy of the DNA, which is going to be used to create the amino acid chain. And ribosomal RNA is associated to form the ribosomes, which we need the ribosomes for protein creation. Here we have our 3 different types. The ribosomal RNA, where we have a ribosome and we have 2 different subunits, a larger subunit and a smaller subunit. Our transfer RNA and our messenger RNA. We have a poly A tail, which is just a whole bunch of adenosine at the very end of the tail on the 3' end. And there's a 5' cap. This cap, it contains 7-methylguanosine. The cap is really just to kind of protect that end to make sure that it doesn't get unraveled from that end. So transcription of RNA synthesis. So the ribonucleotide sequence in an RNA molecule is complementary to the DNA sequence of one of the parent DNA strands, except thymine in DNA is transcribed to uracil in RNA. Let's just repeating that again. Transcription of a gene does not necessarily occur from the template DNA strand. It may be transcribed from the anti-template DNA strand also. It depends on the location of the gene in the DNA. And again, if we think back to the DNA, again, we have both sides of the DNA depending on where it is, and we also have methylation. 70% of our genes are methylated so that they're turned off. So we're going to pick and choose the different chunks and where different things are and where they're going to be expressed, which makes you, you. Okay, so this figure right here describes that genes can be transcribed from both strands of DNA. The arrowhead shows the direction of gene transcription. Red solid bar indicate the gene. For transcription, template DNA strands is red in the 3' to 5' direction. So transcription of DNA into RNA is catalyzed by DNA-dependent RNA polymerase. DNA-dependent, you need DNA in order for the RNA polymerase to work. And it does not need a primer. And this is actually also why the RNA polymerase can be used to make the primer. So we have primase or RNA polymerase when we're talking about DNA replication earlier. So beginning at DNA promoter region, the RNA polymerase attaches to the upstream promoter site towards the 3' end of the template strand of the gene and transcribes. We talked about that, remember, when we talked about methylation. The upstream promoter site is the site where it promotes it to bind. And if it's not methylated before that, then that's going to be open and a gene will be expressed. So that's going to toward the 3' end of the template strand of the gene and transcribes. Sigma factor recognizes the promoter for the RNA polymerase. RNA polymerase binds to the sigma factor at the promoter site of the template to initiate transcription. As the transcription begins, sigma factor is released and recycled. Okay, eukaryotic transcription, RNA polymerase 1. There's a couple of different RNA polymerases, okay? So the RNA polymerase 1 is located in the nucleoli, not the nucleus, the nucleoli. Those are different. If you don't remember your cell anatomy, go back and look what's the difference between a nucleus and a nucleoli. It synthesizes precursors of most RNAs. RNA polymerase 2 is located in the nucleoplasm. It synthesizes mRNA precursors. The RNA polymerase 3 is located in the nucleoplasm, and that synthesizes a variety of small nuclear and cytosolic RNAs and tRNAs. Transcription. This is the template-dependent process where the DNA template is copied from a 3' to 5' direction. RNA chain grows in the 5' to 3' direction. This is where you really want to pay attention and focus on what the question is asking. Is it saying, is the template being copied or is the chain growing? If it's being copied, so the DNA in this case, the DNA is read in a 3' to 5' direction. It reads in a 3' to 5' direction. But the RNA chain grows in the 5' to 3' direction. Do you see the little bit subtle difference there? Reading it 3' to 5' versus making it 5' to 3'. It's slightly different. Just be really, really careful when you're reading questions and see what is it actually asking. So the ribonucleoside triphosphates, or the ribonucleotides, serve as the precursors for the RNA chain. Again, that's the adenine, guanine, uracil, and cytosine. DNA-dependent RNA polymerases. It's going to adds ribonucleotide units to the 3' end of the growing RNA chain. RNA synthesis is initiated at specific DNA sequences called promoter